Couple of big picture things, and then we can get started. So first of all, it was cool to see a bunch of you guys at the Rise Talk on Wednesday. And I had a couple of moments during the talk where I was like looking around the room like, which immunology people are here? Because it was so immunology heavy. So it was fabulous, and I hope that those of you guys who went enjoyed it. And I was excited to be like, look, she's talking about ADCC. Hopefully you're going to be ADCC. But anyway, so it was a pretty cool talk for those of you who were not able to make it. So couple of scheduling things. According to the course schedule, we have a few assignments coming up. I told you that I was kind of working on what I wanted to do with the practical immunology assignment, kind of because of how I had moved some other things around earlier. To be honest with you, the practical immunology assignment originally, when I very first designed all of this, was going to have to do with the dirty mouse paper, which we already did in class. So kind of hard to have a separate assignment about it. And I had another idea, but it just wasn't coming together for me. We also are supposed to be talking about a paper on Monday, with discussion questions. The paper that I want to talk about exists. Can I find it to save my life? No. But I know it exists. In any case, I also know that I'm not going to get through everything I have here today. And I also was looking at the schedule and realized there are a couple things that there isn't an obvious place for that I want to be able to talk about. So I want to make Monday more of a lecture day to both finish some of this stuff and do some other stuff. And I haven't decided exactly how I'm going to arrange that, but we'll see where it goes. And so both of those things are not going to happen. I, of course, did not want to have you guys miss out on the points, because I know a lot of people have the assignment points as kind of helpful for their grade, especially the discussion questions, which tend to be sort of gimme questions or gimme points. And so what I did is I reallocated the points amongst the other things that we have coming up for the semester. So the other assignments are remaining the same in terms of due dates, but I have changed the numbers of points that they're worth. Honestly, they're going to be the same assignments. They're not actually like getting bigger or anything. It's just that way you'll be able to make sure you earn the same number of points. You know, things you did in the past won't suddenly weigh more or something like that. You get to keep the same weights on everything that I told you at the beginning of the semester. These will just have a little more points. I'm planning to post all 4 of those things this weekend. So note that one of them is due the Monday after Thanksgiving. One of them is due the Monday after that. And then the other 2 are both due the last day of classes. I will tell you that the vaccine immunology thing is going to involve like an Excel spreadsheet where you play around with some stuff. And the defining immunity is really going to be like write a page reflecting on this. They're all getting posted this weekend. So you can time them as you want over the next few weeks. So that's the deal with upcoming assignments. I'll also send this to you as in an email, but I thought I should at least let you all know. Today we're going to talk about breaking tolerance and autoimmunity. Like I said, I'm not going to get through all these slides, but that's okay. I know about how far I will get, and there are a couple different ways to rearrange some stuff for Monday, so that'll be cool. And then as we get to the end, I will give you back your exams that you took and talk a little bit about the exams. So last time we talked about mechanisms of peripheral tolerance, and I told you that there were 6 mechanisms of peripheral tolerance. Those 6 were clonal deletion, clonal anergy, clonal deviation, clonal helplessness, and clonal suppression. I don't remember if that was the same order I told you them in before, but that's okay. It's the order that I just thought of them. So the goal today is first to tell you about the 6th mechanism that I didn't get to last time. And then to start talking about ways that some of those mechanisms can be broken. And we can think about how autoimmunity can occur. So some of our mechanisms that you saw in terms of peripheral tolerance last time sort of followed kind of obviously from things that you saw earlier in the semester. To be honest, the sixth one kind of does as well, but it's one of the things that's not going to be it's not necessarily obvious from things you've seen before. In keeping with the theme of everything starting with clonal, the way people usually start this one with clonal is by calling it clonal ignorance. That said, I often, and most immunologists often refer to this one as immune privilege. And so I'll mention immune privilege a lot, but if we wanted to be sort of consistent with our other phrasing, we would call it clonal ignorance. Immune privilege or clonal ignorance has to do with certain anatomic locations in the body. And you can see some of those anatomic locations on this slide. And we need to think a little bit about these organs, and then we'll sort of talk about some details of immune privilege. So if you look at the organs that are listed on this slide, what do they all have in common in terms of, say, evolution? And survival and stuff. Yeah, Kyra. They all make you really fit. Or another way, the way I usually think about it is these are ones that if they got messed up, it would really hurt your ability to survive and reproduce. You would really not be able to have progeny and reproduce and kind of succeed well as a human if one of those got messed up. And so I often think about it that these are things that are like really important not to have messed up when I think about these organs that are involved with immune privilege. As you will see as I talk about immune privilege, there are kind of a couple different ways immunologists think about immune privilege. And so I'm going to tell you the classic way we think about immune privilege, and I'm kind of going to tell you some stories about classic immune privilege. Then I'm going to tell you a little story that has told us that like maybe that's not totally right. And we're still trying to figure out what is totally right. At least in terms of thinking about the classic idea of immune privilege, the idea is that these organs, known as the immune privilege sites, are so important, and it would evolutionarily be so bad if there were problems that we have evolved to exclude the immune system from these organs. We've basically set it up so that maybe with the trafficking of immune cells or other kind of access of immune cells, we're just like, nope, you can't come in. There's a wall so that those immune cells cannot come to this organ. And the idea is that that will allow us to avoid damage to those organs, because you can never have the immune system make a mistake there and start killing things there. It's the classic idea of immune privilege. I'm guessing I know what your question is because there's a sort of obvious question from this. Yes, what's your question?
I was going to say that, let's say like there's some parasitic hosts that can go into these.
Mm-hmm.
What are ways of applying protection?
We'll talk about that later. So yeah, there are some places where you're like, I see some flaws in this plan. We're going to get to the flaws in the plan. Don't worry. But right now we're going to pretend that the classic definition is perfect. And in every immunology textbook, there is discussion of this one particular disease state that is sort of the classic disease state that everybody thinks about with immune privilege. Also, students often like to ask me questions about this one. This reminds me of one important point I should make about this time in the course. Usually I think I make it like around here, which is, remember, as we talk about clinical immunology topics, I am a PhD in immunology. I'm not an MD. And so suddenly people will start to ask me like really picky clinical questions about certain things, and I haven't been trained in that. In particular, I definitely, so like, I kind of know how allergy medicines work, for example, but for example, I cannot tell you why your doctor chose that particular allergy medicine for your prescription, because I'm not an MD and I'm definitely not your MD. So, know that sometimes throughout the clinical parts, you may have questions that are not things I'm able to answer. I say that because there's also some questions that often come up with this case that I haven't figured out the answer to. And I agree that sometimes I'm like, but it's like the classic case that everybody always talks about. And it is a disease called sympathetic ophthalmia. One fun fact about sympathetic ophthalmia is that sympathetic ophthalmia is actually the reason why Louis Braille, the guy who made up Braille, went blind. So he had sympathetic ophthalmia and went blind because of sympathetic ophthalmia. So like, it's important and real and stuff. And sympathetic ophthalmia involves the eye, which is one of those immune privileged sites. And remember that the idea, the sort of classic idea of the immune privileged site, is that we completely exclude the immune system from that organ. Another way you might think about that is that basically like we're hiding that organ and the antigens from the immune system. So we don't let that immune system ever see that. And so we've got the eye, this nice hidden area from the immune system. However, if an individual has trauma to an eye, like maybe their eye gets stabbed or something, that might break that barrier that was keeping the immune response away from the eye, or that was hiding the eye antigens from the immune system. And so now when we have this trauma to the eye antigens leak out of the eye and go into local lymph nodes, a local secondary lymphoid organ. Previously, these antigens have not been around activating T cells, and so we don't see much T cell activation. And now when these antigens make it to the lymph nodes, the T cells are like, oh my god, look at this new foreign antigen. There's a whole bunch of it. This is very scary. We gotta do a thing. And those T cells will go back to the eye and start attacking the eye and killing the eye because now they found this new antigen and start killing the eye. But not only that, the T cells will actually go kill the other eye too, because now there's a second source of this new quote unquote scary sounding antigen. And so even though our patient has only had trauma to one eye, they end up going blind in both eyes because of immunologic destruction of both eyes after those antigens have come out of being hidden. Yep, Grace.
Were the T cells, like, do they not recognize it from when they were tested in the thymus with all the proteins, whatever?
It's a great question. This is the very classic definition of sympathetic ophthalmia. As I've been explaining it, I've like been like sitting here, but what about this? But what about this? Even in my head as I'm standing here talking. At least one of my but what about this is, I actually came up with an answer that is one of the later slides. So I think that there are a lot of sort of like exactly what's up with this questions. I will give you part of my answer on that one is that when you have that trauma to the eye, you're also probably inducing a fair amount of inflammation. And so some of this may be related to having that inflammation along with the antigen. But again, the classic answer is just, it was hidden and now it's not. Okay. My guess is that inflammation coming from the trauma is probably pretty important here too. So this is kind of, like I said, the classic version of immune privilege. One other sort of classic way that we can think about immune privilege on a slightly different scale as well is that sometimes when we think about certain autoimmune diseases, we talk about a process known as epitope spreading. It's also referred to sometimes as autoimmune amplification. And the idea is that perhaps we might have some antigens that are really, say, intracellular antigens, like green square and yellow triangle. And after we have an infection, those might get released outside of the cell, and maybe at least in the case of, say, B cells and antibodies, before those were hidden, now they are available. And so now we have more epitopes that we might start to respond against because those previously hidden antigens have now not become hidden. They've become exposed as the cell died and they were released from the cell. And so this is sort of a sort of related thing about like the idea of a physical barrier that's holding some antigens from being recognized. And if that barrier is breached, then and the antigens are recognized, then we can start to get a response. So again, if we just think about the classic definition of immune privilege, you start to be like, I don't know what's going on here. Or at least you start to ask some questions. And so I want to tell you kind of two directions that come from that. And so here you can see I'm listing this as problems with immune privilege. One of the reasons why I have the slide set in this way is because I wanted to remind you what the key immune privilege organs are. And I say that because I'm going in both of these kind of stories I'm going to tell you. The organs are going to be important. So I wanted to remind you what organs we're talking about. So one story is kind of a broad story, and it is related to Ebola virus. And basically the way I think about this story is some of the things that we've learned that are related to Ebola, particularly after the 2014 West African Ebola outbreak. So before that, so this is a graph that shows some information about Ebola outbreaks before that West African Ebola outbreak. Specifically because this was published in 2007, these were the epidemics of Ebola that had happened before 2007. What you can see here are years on the x-axis of different outbreaks, and then number of cases in the outbreak on the y-axis. In the darker color are the number of people who died of Ebola in each outbreak, while the total bar with the lighter color is the number of people who were infected, and then the number is actually telling us the overall number who were infected. So there are a few things that you should kind of be aware of about Ebola knowledge before 2014, which are brought up in this plot, though I could talk about this plot for a very long time. I'm not going to say all the things I could tell you about it. One of the things you should notice is that the outbreaks are super sporadic. They're not like happening at regular intervals. Like the virus just like goes away for a while and then comes back. And that's a hot topic in the field. And so one thing to know is that this is kind of like sporadic. The locations are a little sporadic, that kind of stuff. So that has always made some of the study of this really challenging. But the other thing, and the main reason why I'm showing you this, has to do with the numbers on most of these bars and sort of the general way that these bars look. So if you look at this before 2007, and it remained true until 2014, what was the largest ever Ebola outbreak at that we'd ever seen in the world at that point? Yeah, Andrew.
2007.
Okay, 2001, which was how many? 425 people were involved. When you think about those 425 people, it doesn't matter for us that we're exactly perfect on this number, but about how many people died? and how many people lived? Yeah, Kyra. Or Emma. Your name's Emma. Your name's Kyra. You both raised your hand. About half of the people died, half the people lived. So there were like 200 survivors. So like in the biggest ever time of there being survivors of Ebola, there were 200, right? Most of the time there's way fewer than that. Like it's usually a really small number of people that live through an Ebola epidemic. And so there were always a few Ebola survivors. To contrast this, here are some data about the 2014 outbreak. I know this is hard to read. I know some of you guys have it in front of you. Again, we can just talk about the reason why I'm showing you this, or the point that I want to make. We're looking at different countries here. We have number of deaths in darker blue, number of cases in lighter blue. So it's basically the same type of thing I was showing you for the earlier outbreaks. And the key thing I want you to look at here is the total. So what were the total numbers in terms of deaths and cases in this West African Ebola outbreak? Okay, so Andrew's like, it's a lot. What are those numbers specifically? Yeah.
So the total here of cases in total is around 28,646.
Mm-hmm.
And the death is around 11,000.
Yeah, so there were 11,323 deaths, and there were 28,646 cases that were reported. So much, like compared to the previous high, which was 425, and so this was just like so much bigger than ever before. But come on. But the particular thing I want you to notice about this is how many survivors there were. There were like 17,000 people who lived through this Ebola outbreak, but who got infected with Ebola. 17,000 Ebola survivors is, as far as we know, more than the number of Ebola survivors in the history of the world before that. Like one time there was 200, and that was like shockingly a lot, but usually there's like 5, right? So I mention this because for years in all of those previous Ebola outbreaks, there was sort of this like story, there was sort of this like almost anecdotal information that if you survived Ebola, you had other health problems. But no one could really like figure out a lot of the details in the data. And part of the problem was like, cause the study population was like 5 people. And so if one person had a thing, is that cause they were one random unlucky guy? Or is that cause 20% of people who survive Ebola get that thing? Like the numbers were just so low that it was hard to know if this was like weird random like hearsay stories or if it was a thing that once you had Ebola, you had other health problems. And after 2014, suddenly there were all these Ebola survivors that we'd never had before. And when we kind of started thinking about what was going on and really studying them, some weird things happened. The first case, the first person that people started talking about was Ian Crozier, who was a doctor who was, I believe he was a British doctor, who was in West Africa treating patients. He got Ebola pretty early because he was treating patients before they had PPE, to really be treating the patients. So he got Ebola. Fortunately, he, ended up, being cured. But then like months later, his eyes changed color. So his eye had previously looked like this, and it started to look like that. And like, that's super weird, right? And when people started to try to figure out what was going on, they realized that he actually had been cured of the virus from his whole body, except for his eye. And the virus had been living in his eye all that time, and had caused damage in his eye such that this happened. That's super weird, right? So this mentions sort of some of the kind of ideas about previous outbreaks. People have actually now published some of these questions about long-term changes in Ebola survivors. But what I want you to notice here, and so here you can mention, there have been these consequences, but nobody really knew what was up, is what it mentions up here. But now the results show that like there is a significant increase. And these things include eye problems, like pain or blurred vision, loss of hearing, sleep difficulty, joint pain, abdominal and back pain, fatigue, impotence, severe headaches, memory problems, and confusion. If you look at all of those symptoms and think about the person I showed you before, what might you notice that all of those symptoms and his situation all have in common? Yeah, Emma.
Most of them involve the head region, so like ears, eyes, brain.
Most of them involve the head region, particularly eyes or brain. Even the ear ones you realize are really kind of brain related. You see a lot of things with eye and brain. Why might I be pointing out eye and brain here? Yeah, Kyra. Those are two of our major immune privilege sites. And so Andrew had a question when I first described immune privilege. What was your question, Andrew?
So, any other virus or parasite live inside of these immunological?
And so what we are starting to think might go on with Ebola is that your immune system might be able to get rid of this virus from everywhere in your body except for the immune privilege sites. And it may just hang out in the immune privilege sites and start to cause some of these issues. And so exactly the issue that Andrew's talking about may be the case. Maybe it's only true in, some people with some other things going on with them. Again, we still don't really understand a lot of this, but it's really interesting to see all of these Ebola problems seem to relate to the virus being present in immune privilege sites for longer periods of time. On a sort of related front, with Ebola, there were a few cases of Ebola that seemed to be sexually transmitted. And there are this is again still a bit debated, but it does look like Ebola can be transmitted in semen. In some cases for like a year or more after a person is quote unquote cured. Again, that's one of our immune privilege sites. And so there may be some question about whether or not Ebola is spending time in yet another immune privilege site. This has led to all sorts of epidemiological consequences. Poor Pauline Cafferty actually ended up getting Ebola 3 times during the 2014 outbreak because she got it. She was cured. It came out of one of her immune privilege sites and she got sick again. She got cured, came out of one of her immune privilege sites again. She was a nurse treating patients. She got sued for coming back to work with Ebola. It's been a whole sort of medical fiasco. In addition, there have been a couple of cases where we thought that different areas were Ebola free. So usually if people in that area test positive or test negative for, I believe, 42 days, then we say that the area is Ebola free. There were some areas where it was like negative for months. And then all of a sudden there were like 1 or 2 cases. And it turned out that they were sexually transmitted cases from this persistent virus in immune privileged sites. One of the really long cases was actually a woman who had been pregnant for part of the time. And it was a whole thing because then she spread to her partner. And it was a big sort of problematic case. And so this is one example where we're starting to be like, maybe this virus does have something going on where it's actually taking advantage of this immune privilege site. We don't have a ton of examples of this. And there are lots of reasons, and I don't want to go into like, virology is its whole own class in the spring, so I'm not going to like start like geeking out about why certain organs are potentially important. But one thing that we're starting to wonder about is whether particularly Ebola with some of this evidence may persist in immune privileged sites because of that lowered immune response or that lack of immune response there. Yeah, Emma. Yeah. Okay, which? This one?
Yeah. Was there a reason why there were like a proportion of survivors in Sierra Leone and Liberia seemed to be higher than in Guinea? I mean, in Guinea it seems like 75%-ish of people who had Ebola died, but with Sierra Leone and Liberia it seems to be less than 50%.
So, yes, that was one of my classes last spring, was emerging infectious disease. There are a bunch of different reasons that we talk about. And often when I actually talk about this graph there, I also point out Mali. Whereas if you look at the numbers, it's, dramatically high in terms of fatalities there. And so part of this is going to be related to, healthcare availability, and how well people are getting care. Some of this is also due to diagnostics. One of the things that is really nice in, say, Sierra Leone is that they were testing a lot of people. And so people who had, minor isn't the right word, mild disease, got tested and counted as positive. It wasn't like the only way you got counted was that you died. And so in some cases, the only people who were testing positive were the deaths or like the people who were really severely injured because they didn't have good diagnostic capacity. They couldn't diagnose the more mild cases. And so some of this is about sort of diagnosis and reporting issues, and some of this is about healthcare availability is really the biggest issue. Yeah, Andrew.
So let's say that the eye, for example, say if a virus does affect the eye, and then what's inside it? Can a virus proliferate and like grow
it possibly?
So is there any way to like combat that once you know that the eye is infected? I know it probably goes deep into
it does go deep.
Yeah.
It does go deep. Yeah. So can I do the other example and then come back to that story? Because part of my answer, I have two answers and one of them is related to the other example. So that's why I want to do that one so then I can segue. So like I've mentioned, everything we've been talking about here has really been kind of the classic idea of immune privilege. And there were always kind of, and you all are already correctly pointing it out, places where you're like, but, you felt a little uncertain about the idea, but like sympathetic ophthalmia, that's what happens. This is a thing. And there are a few ways in situations where we've realized that it probably doesn't work quite as, cleanly as we might have thought in that classic model. And the most clear example of this, the place that really again, I could tell you tons of data about why we're but there's one really good example of us being immune privileged, not quite like that. And that has to do with some details regarding multiple sclerosis. So we'll see more about multiple sclerosis going forward. But what I want you to know about multiple sclerosis is that typically your neurons will have this myelin sheath, which is basically a lipid that is protecting that sheath or that thing and allowing electrical impulses to go in the sort of correct direction, and not kind of try to short and go out of the neuron this way, to not try to come here, but to make the signal go like this. It's honestly the same thing as the plastic coating on your wires, where if the plastic coating starts to come off, you get little sparks coming out the side. It's because the electricity is trying to go this way instead of going this way. Myelin sheath is doing exactly the same thing. In multiple sclerosis, there's destruction of the myelin sheath. So you can see there's little holes coming into the myelin sheath. And so electrical impulses are not able to transit correctly in our patients. And multiple sclerosis has a lot of parts of the immune system that are important, but one really major important part is that there are T cells that actually recognize myelin antigens and lead to destruction of the myelin sheath, killing of cells that make myelin, destruction of the myelin sheath. We end up getting lack of myelin because of these T cells that are attacking in the brain. And we can say more about multiple sclerosis at a later point, but that's kind of what you need to know about multiple sclerosis at this point. And when people sort of heard and thought about this, they said, aha, I figured it out. I figured out what's up with MS. The problem is that the brain's immune privilege somehow got messed up. So we used to have it so that T cells couldn't go there, right? And now the T cells are going there somehow. So what we've got to do is we've got to figure out a way to stop the T cells from going to the brain. And then it's like we reestablish the privilege and we all live happily ever after. Or we at least stop the damage. That was sort of the idea. And so some immunologists made a drug, and that drug is known as Tisabri, and it is an antibody that specifically blocks an integrin. And as a result, T cells can do rolling and chemokine select processes, but they can't finish their trafficking. And this antibody is specifically against the integrin that T cells need to use to go to the brain. So the idea is like now we're just going to stop T cells going to the brain. We're going to almost like reestablish that privilege so the T cells can't go there and mess things up anymore. And we'll all live happily ever after. This drug was absolutely revolutionary. So in the blue line, you're looking at time and you're looking at number of new lesions that could be seen on the brain in MRI, because the T cells will keep destroying parts of the brain, and so you'll see more and more lesions. And so blue is kind of what we often see in a patient, where we just see increasing numbers of lesions as the T cells are destroying more and more places. And when patients were treated with this drug, you can see that basically the further damage was stopped. Yeah, they still had whatever kind of damage they had when they started, but we basically stopped progression of that disease. Like revolutionary information here. The Nobel Prize in Medicine is given every year, the first Monday in October. And when you are an immunology or biomedical sciences nerd like me, you find out that actually there are websites that take bets on who the winner is going to be, and there's like a short list of names that people talk about every year. And the person who came up with this is always on that short list. However, when Tisabri was introduced, and when it was approved in 2004, there was a problem. And what you can notice is in 2005, this drug got pulled from the market because of two patients who had a really severe disease after taking this drug. And this disease is called PML. And you can see there was sort of back and forth, goes on the market, comes back off the market because of PML. You can see, a bunch of patients die because of PML. And if a patient doesn't die, PML is a very severe brain disease with demyelination and a bunch of brain impacts. PML stands for progressive multifocal leukoencephalopathy. So we probably are like, okay, what's up with PML? What's going on with PML? It turns out that PML is caused by a virus. It is called JC virus. I'm forgetting it now, and I only learned this like within the past year. I've always known PML is caused by JC virus. I actually worked with somebody in like 2002, 2003 who worked on this virus all the way. I knew the person who was like the expert on JC when this was happening. And beforehand, we were always like, why do you work on this virus that no one cares about and like 3 people in the world get? So I've heard about it forever. I just learned like within the past year, JC is like stands for the person's initials that it was first like came out of. So I think it's like John Cunningham virus or something. I can't remember the exact name, but something like that. In any case, when people started to look, turns out that JC virus is in 80% of people. It's in most people, causing absolutely no problem. It's entirely possible that some of you could have JC virus and have no knowledge of it. It's causing you 0 issues. And JC virus infects the brain. And so in most people, JC virus gets into the brain, infects the brain, and you actually get an immune response that gets rid of JC virus in the brain. But if you block T cells from going to the brain, suddenly that virus starts to destroy your brain. And that virus runs rampant because the T cells can't go deal with it anymore. And so, in fact, when they started putting Tysabri back onto the market, a big part of what they would do is test to see if patients had JC. And the only way you could get Tysabri is if you didn't have JC. But this also kind of impacted our understanding of immune privilege because we went from saying like, oh, the original idea is that no T cells in the brain ever. It just is that somehow that got messed up in MS. But now if we actually block T cells from going to the brain, we see that we kind of have viruses that can go out of control. And so it looks like in the normal healthy brain, there is an immune response. There are some immune cells going on. And this is probably true, Andrew, also in the eye. There probably really are some immune responses happening. We used to be like, there's none. But now clearly there have to be some in the brain. So what we think now really is what's going on with the immune privileged sites is that the immune privileged sites have a more immunoregulatory environment. So maybe they are making a lot of IL-10 in that area or other things to keep the immune system kind of calmed down. We allow the immune system in, but we put really tight controls on it, and we have kind of an environment that's really immunoregulatory. And so usually we're kind of trying to keep the immune system quiet there so we can't have immunopathology. It may not be as much of a just keep it all out the way we normally thought. And so that might be part of the sympathetic ophthalmia question related to what I was saying to Grace before. Maybe some of it is that the trauma, the inflammation from the trauma breaks that quiet environment. So it's both the antigens are around and you have no more quietness. And so you get rid of that immunoregulatory environment. So we're still trying to figure that out, but we often talk about the immune privileged sites as one of the mechanisms of peripheral tolerance. So, of course, autoimmunity results from a failure of tolerance. And as I mentioned to you before, that can be due to some kind of genetic factor, or that can be due to some kind of infection or environmental exposure. And all of these things are going to impact our mechanisms of tolerance. And so you might now, we saw these data before about common autoimmune diseases. And before we saw them, we were thinking about all the places where central tolerance could go wrong. And we were like, oh my gosh, how are autoimmune diseases so rare? They should happen like all the time, 24/7, given all those central tolerance problems. But now I've shown you 6 different layers of things that can protect you. And now you're kind of like, these might be a little more common than I feel like they should be. If we got all those different redundant fail-safe mechanisms. And so we need to think about how some of those fail-safe mechanisms can go wrong. We can see problems in both central tolerance and in peripheral tolerance. The disease states can look a little different when it's a problem in central tolerance versus a problem in peripheral tolerance. So I'm going to tell you about 2 disease states that come up that are really defects in central tolerance. And we'll kind of talk about what's the overriding feature of autoimmune diseases that are central tolerance problems. And then we'll move into how the heck peripheral tolerance can get messed up. One of our disease states that is a problem with central tolerance is known as APESED, and I've actually already mentioned it earlier in the semester. So APESED stands for autoimmune polyendocrinopathy and candidiasis with ectodermal dystrophy. And in APESED, the patients are actually missing air, so their thymus does not make all of those tissue-specific proteins. And so we can't delete T cells that are highly reactive to tissue-specific antigen. And so they make their way out of the thymus, and they cause autoimmune diseases in a number of different organs in our patient, as well as a bunch of different other symptoms. And so the key thing to note here is when these patients have a defect in central tolerance, they don't have one autoimmune disease or one autoimmune symptom. They have systemic autoimmunity where they have many autoimmune diseases coexisting, because the problem is sort of a general problem in getting rid of autoreactive cells. It's not that there was like just the one cell that slipped through the cracks. It was that we're letting out a lot of cells, and so we're going to end up with a lot of autoimmune problems. For reasons that we don't totally understand, it seems like endocrine glands are often really targeted in all of this. The other really famous defect in central tolerance is a disease known as IPEX, which stands for immune dysregulation polyendocrinopathy enteropathy X-linked. It's because it's an X-linked disease. The gene is on the X chromosome, so it's mostly found in boys. You can see that, again, there's something going on with endocrine stuff. Endocrine glands seem to be attacked pretty often. And there's many polyendocrinopathy. And this is actually a genetic defect in FOXP3. So in the boys who have IPEX, they are not making FOXP3, and so they can't make Tregs. And so they've basically lost all Treg production. You can see that this manifests with autoimmune enteropathy, which is issues with the GI tract, eczema or other dermatitis, nail dystrophy, autoimmune endocrinopathies, so something with the endocrine system, skin conditions, enlargement of secondary lymphoid organs, diabetes, food allergy, infections, and most of the kids who have this die within the first 2 years of life. And so again, what I want you to notice is with a defect in central tolerance, we have a really broad set of symptoms where we're getting autoimmune diseases attacking many, many, many antigens, many, many, many organs, because it's not just the one cell that went through the cracks. For the rest of the time, we're going to spend some time talking about ways that peripheral tolerance can go wrong. There are a bunch of ways that we can think about peripheral tolerance going wrong. I'm going to tell you specifically about ways that clonal deviation and clonal anergy can get messed up. I'm not going to tell you about how clonal deletion can really get messed up, because that would mean that we're bringing cells back from the dead. Not really a thing. I'm also not going to really tell you about how helplessness gets messed up, because in some ways that's also bringing cells back from the dead. Still not possible. I'm not totally mentioning privilege getting messed up, because you can see there's a lot of things we still need to figure out about privilege or clonal ignorance. And clonal suppression, I'm actually going to throw in with deviation, because remember, clonal deviation was about making like Th1 versus Th2 and how you push the T cells in different directions. one of the ways that things go wrong with clonal suppression is really the same thing, that you should have been pushing to Treg and you pushed to something else instead, like a Th1. So I'm kind of going to be throwing the breaking suppression in with breaking deviation. All of the examples that I'm going to give you are really examples that tend to relate to infection outcomes. And so a lot of them have to do with infections being related to autoimmune diseases. That does not mean that every single autoimmune disease is related to an infection, but the examples that we understand tend to all have this happen. And you can see some examples from your textbook here, where we can see that some people infected with particular organisms that also have a particular HLA type may end up with a particular autoimmune disease. And so what that should tell you is that the microbe is particularly important, but there's also probably something about which antigen is being presented, because it's only present in people of certain HLA types, in order to make this occur. And usually none of these are like 100% situations. It's not like 100% of people who have HLA-B27 who get chlamydia trachomatis will end up with this arthritis presentation. That math and that sort of the fact that none of it's 100%, and so it's like, well, we'll say 5% of people have B27 and 5% of that 5% get chlamydia. And then 5% of the 5% of the 5% get autoimmune disease. The math makes it look really like random and sporadic. It's actually very hard to tie these things together. One of the things you need to be able to do is like, oh, hello, you have arthritis. 20 years ago, did you have chlamydia? Like, some of these are things that you might be able to say, yeah, I had that in the past. Some of them are like, I don't know if I had it when I was 2. Like, these are just random things. So we don't necessarily know what you were infected with in the past. And that might have been a long time ago, so we can't tie it with stuff that's going on. And so a lot of these associations are really, really hard to do. People are still talking about them. There is, one of the papers I was thinking a lot about, talking about, it's such a cool story, but it wasn't like the paper wasn't like good for discussing. Has to do with multiple sclerosis and some data on whether having Epstein-Barr virus infection can predispose one to multiple sclerosis. And it's been kind of rumored in the literature for years. And there are like hints here and there, but it was never like really clear, because like 95% of people get Epstein-Barr at some point in their life. So like, but 95% of people don't get multiple sclerosis. So clearly there's like other things going on and what's up. There actually is an amazing paper. So there are a couple really good papers that are showing kind of a cool immunological mechanism, how that might be true, though it might depend on like when in your life you're getting it and things like that. But the paper that really, in my mind, like showed the association was a paper that involved data, going through and mining data from U.S. Army recruits over years and years and years, because the Army keeps data on everyone and actually tests them for EBV when they join. And so we actually knew, did you have EBV before or after, kind of thing. Just to give you, I can't remember the exact number, and it's so cool that I want to find it. I was looking at it last night, so I should be able to find it pretty easily. Okay, in order to do this study, they had to look at 10 million people and 62 million samples to get enough data to statistically make the association and to figure out how to work. Like, this study is shocking in terms of how much data they had to analyze, but it actually shows, like, comes out with the stats of it pretty well. but no one has ever looked at 62 million samples before. And so, in many of these cases that I'm mentioning on this slide, like, we sort of hypothesize what's going on, but we have not done a study of that ridiculous size, to actually be able to sort of nail it down super well. But all of my examples are going to be about infections and breaking tolerance. So the first example that I want to give you is a clonal deviation example, kind of a clonal suppression example. And so the idea here is that in a normal sort of healthy situation, when a particular T cell is activated, that T cell should receive polarizing cytokines to become one particular kind of T cell. In this Helminth slide, this Helminth example, ideally we want that T cell to become a Th2 cell. Good things happen. One lives happily ever after. But if it just so happens that the T cell gets activated to make a Th1 response, then suddenly that T cell may actually do things that are damaging during Helminth infection instead of protective. And so we might think about, well, why would it be that the T cell might make a Th1 response when it was supposed to be making a Th2 response? And one way you might imagine that is that perhaps other polarizing cytokines were being made at the same time. And so the T cell was supposed to only see IL-4 and get pushed towards a Th2 response. But maybe because you were really unlucky and the day that T cell got turned on, you also had another infection, that T cell happened to see some IL-12. And that T cell happened to get pushed towards the Th1 situation. And so just because you were unlucky, that T cell was in the wrong IL-12y place at the wrong time, it turned into a Th1 cell. And so now you got a problem. We've started to understand this specifically in a couple of specific cases related to two diseases that I'll show you here. And so this is one paper looking at this with a relationship to Crohn's disease. And so in this study, there were some mice who were wild type mice. And those mice got infected with a virus. Two different versions of the virus that had different types of genetics. And if the wild type mouse got infected with either the blue version or the red version, the mouse was a little sick from the virus, but then was fine. However, if the mouse had a change in its genetics and got the right genetic version of the virus, so the mouse had to have this particular type of genetics and this particular type of virus infection. So again, it's a little bit unlucky. That mouse may have its immune response pushed towards a Crohn's disease situation. And so what you can notice here is in this situation and in the one I'm going to show you on in the next slides, there's both a kind of genetics thing and like a pathogen thing and a little bit of a timing thing, especially in the other one. And so sometimes when people will ask me like, well, why did I get an autoimmune disease? The answer is kind of like, because you were really unlucky that you had the right genetics and the right microbe and the right timing. Like you got struck by 3 bad lightnings. And that nobody likes that as an answer. Like nobody feels like, yes, thank you for answering that. Many of these things are also really hard to figure out. This sort of goes back to what I was saying about the infection thing, because usually what happens is when you turn on that first autoreactive T cell, you don't start to have autoimmune symptoms like that day. You need some time before the organ gets damaged before you're actually going to go to the doctor. And so tying back that infection that happened a long time ago with those symptoms which might be with a totally different organ, is really hard. So the other example that we're really are starting to understand better is an example that is related to celiac disease. Celiac disease is a little bit complicated in and of itself. In celiac disease, we have an antigen, which is a particular part of a carbohydrate. So we'll just say gluten. Normally, we eat this antigen. I eat this antigen all the time. I love carbs. Normally, I eat this antigen. It comes into my GI tract. Dendritic cells in my GI tract pick up this antigen. There's no inflammation. That we will present that antigen to a CD4 T cell. That CD4 T cell will become a Treg because we didn't give that T cell any other cytokines. It only got sort of a tolerogenic set of things. Dendritic cells in the GI tract, if they're not, pushed in any other direction, are going to induce tolerance because we don't want to be allergic to every kind of food we eat. So the GI tract tends to push things towards tolerance. But if you are unlucky and you happen to get infected with a particular virus the first time you see gluten, suddenly those dendritic cells are going to be doing two things. They're going to be presenting gluten and they're going to be presenting virus antigen. And now when they're doing that, they're going to be making the cytokines as a result of PRR stimulation from the virus. And so now instead of telling that gluten-specific T cell be a Treg, they're going to be making other inflammatory cytokines like IL-12, and they're going to tell that gluten-specific T cell to become a Th1. And when they see gluten, to start attacking instead of to start suppressing. This, of course, has to happen kind of the first time you see gluten. If it happened the second time you see gluten, well, your gluten-specific T cell would have already been polarized to a Treg. It wouldn't get unpolarized to something else. It would have already been a Treg. But if you're unlucky enough that you happen to have the right virus and the right MHC, on the first time, the gluten-specific T cell happens to be in the gut, seeing gluten. Because remember, the T cells are going around the body. If the gluten-specific T cell was here when all this was happening in your GI tract, then it's not getting turned on. It had to happen to be in that location, at the exact same time that that virus was there. And you had to have the right MHC to see the right peptide, and you had to get the right virus. All those things happen. The T cell gets turned into a Th1 when it was supposed to have gotten turned into a Treg. And now you end up with celiac disease where you wouldn't have otherwise. And so you can see this here. Typically, gluten is coming into the cells and we're getting kind of a tolerogenic response. But if gluten and this particular virus both happen to be present at the same location at the same time, and it's the first time you're seeing gluten, or at least the first time this T cell is seeing gluten, so this T cell can get pushed. It's naive, it hasn't been pushed yet, so it can get pushed in a particular direction. It hasn't already been energized. If it already saw something once, it might have been energized and to be totally off. So it's naive. First time it's seeing gluten happens to be at the same time as seeing this virus. So that a dendritic cell can actually push that cell to be a Th1 cell. Now you have this gluten-specific response that's going to cause damage. And so again, you can see that there's a certain amount of unluckiness of you got to have the right virus, the right MHC, the timing of you had to have a naive T cell in your GI tract that day that responded to gluten kind of thing. All of those things had to happen, but if those things happened, then you're going to unfortunately break this tolerance. And we will see the other big way some other big ways that tolerance can be broken, all of which are related to anergy. next time, as well as going into some other details about autoimmunity. So I will see you guys on Monday. You don't have anything due on Monday, but I will email you when the other assignments are posted, and this is the update for new assignments. Oh, right, exams.